As the global pandemic continues, many studies have come out every day trying to explain what is happening around the globe, how this new virus emerged, how it is spread, if it can mute, and as my final project in evolutionary biology, I would like to talk about the evolutionary analysis of coronavirus, especially on SARS-CoV-2 and predictions of possible future scenarios. This presentation will have the purpose to analyze the evolutionary path of SARS-CoV-2 and think of future possible scenarios where the virus could evolve. As for methods, for this project, only scientific articles were used, and the literature review was conducted by using a broad range of search terms like coronavirus, SARS-CoV-2, and evolution. Now I'm going to explain some background genetics. RNA viruses do not have the machinery to correct errors during replication as in the case of DNA-based viruses, but there is an exception to this rule, the coronaviruses. This fact is really important because it means that within populations of RNA virus, we are going to find a big diversity thanks to the lack of error correction machinery. But this comes with a problem. The greater the virus diversity, the more difficult to find a vaccine or treatment. As we mentioned, coronaviruses are the exception to this rule, but that does not prevent these viruses to get affected by others because we can have a coronavirus evolutionary process through homologous recombination. That is, if there are two or more RNA viruses that are infecting a cell, there is the possibility of generating diversity by recombination. To understand the genetics of coronaviruses, it is needed to know that not only point mutations affect but also recombinations. And many scientists are almost sure that SARS-CoV-2 may be the result of a recombination between two very alike viruses inside of a cell. Moving on to the evolutionary origins, RNA viruses are present in protists, algae, plants, fungi, animals, and as eukaryotes came in evolution long after prokaryotes, it is inferred that RNA viruses appear long after the origin of life, and even to date, no RNA viruses have been discovered in archaea, which suggests that RNA viruses are probably not that old. As for classification, there is a strong difference as to how evolutionary biologists and physicians refer to viruses. Often, the latter refer to viruses as pathogens, but considering viruses only as pathogens has led us to classify them according to the type of capsid they have or according to what they affect. For example, human viruses, dog viruses, the organs or systems they affect, viruses of the respiratory system, and so on. This has created a barrier favoring the idea that each virus is a pathogen that arises independently, and this can greatly limit our search for a medical solution. Replication. When the polymerase is reading the RNA virus genome, it will make mistakes that cannot be corrected because most viruses don't have an exonuclease. This will result in mutations, and the larger the genome, the larger number of mutations will occur. Sufficient to say, the number of mutations will be directly proportional to the length of the genome that is being replicated. In the case of coronaviruses, they evolve more slowly than other RNA viruses. Typical polio or influenza virus evolve about 10 to the power of 6 times a million faster than DNA double helix viruses, and instead coronaviruses mutate on average to 8 to 10 times slower than other RNA viruses. But why is that? Coronaviruses are the only known RNA virus that have an exonuclease, as we have mentioned before, that will correct errors in their genomes, and because they can correct errors, they do not accumulate mutations proportionally to the size of the genome, resulting in coronaviruses having huge genomes compared to other viruses. Typically, the genomes of RNA viruses are 12,000 or 13,000 nucleotides, while the SARS-CoV-2 genome is more than 29,000 ribonucleotides long. As discussion, coronaviruses, by having the exonuclease, allow a huge expansion of their genome, and this implies that the large genome is associated with a much more complex expression of its genetic content, which makes them more genetically stable. This is good news because it means that they will not vary as much from patient to patient or year to year, and that it will probably not see resistance to antivirals anytime soon. To conclude, SARS-CoV-2 is a very stable virus. No mutant that is more aggressive or infective than others is currently known, which does not mean that it will not exist in the future, but we do not know of any mutant like that to date. The virus does not stop evolving. It changes a couple of times a month with point mutations, but it will be more appropriate to pay attention in the immediate future to the possibilities of recombination. It is not possible to predict the appearance of a new virus because biological evolution is a multifactorial process that depends on the variability of biological populations, on natural selection, on different selective effects, and this means that it is not possible to predict which mutations they are going to look at a population. What can be anticipated is that sooner or later, in any virus, and especially in RNA viruses, there will be the emergence of mutants resistant to antivirals. These are my references for these presentations, and thank you for your attention.